Okay, thank you so much, Maria. And it is my pleasure today to talk about the work of my lab at the NIH and using deep learning to predict disease causal regulatory variants. And please let me start by going 20 years back. At that time, a good friend of mine, Marcelo Nambrega, decided to go on a crazy mission and deleting the longest intergenic intervals from the mouse genome. So he chopped 2 gene deserts out. And it was quite shocking, at least at that time, to observe that the mice were healthy, viable, happy, if you may say so. And according to over 100 biochemical tests, they were just not. Arguing for vast amounts of junk DNA in the human and supposedly animal genomes. So it might came as a shocking observation that it's not all non-coding DNA in our genomes that junk. We have very specific single nucleotide mutations that we now know are strongly associated with diseases, as for example, this one, GTC variant that's associated with polyadactyly. It's a mutation, a megabase away from the target sewing hedgehog gene and the ZRS enhancer. And such mutations have been associated with tons of different diseases. You go with type 2 diabetes, obesity, Parkinson's, cancers, etc., etc., etc. What is interesting is that when you look at GWAS overall, as you all probably know, the vast majority of GWAS variants are non-coding in nature. So how can we really look at these variants and figure out what they do? So this one, for example, what is the mechanism of regulation? And as you can see, there is a suppressed distal enhancer of alpha-synuclein gene. And when you have a mutation, A2G mutation, what happens, the binding of this protein complex, MX2 and KX6.1 is disrupted, the enhancer is reactivated. Alpha-synuclein is expressed at a higher level, and that increases the risk of Parkinson's. So what my lab is interested in reality, is kind of looking through the sequence of the human genome, stumbling upon the sequence variants, and asking a question, does it appear to be an enhancer? Does it appear to be in a binding site? And if so, does it disrupt binding of a particular transcription factor? And does it have a phenotypic effect on the enhancer activity? How do we do that? We use deep learning, I guess. Who does not, right? And we use a two-phase deep learning model. Step one of phase one, we take all the ENCODE and NIH roadmap epigenomics data, and we model regulatory genome starting from DNA sequence segments. So we scan the genome using these two key sliding windows. Phase two, using this regulatory data, we try to model enhancers across many tissues, 44, 47. And the results are, so this is the work of Sanjar, Hudayberdiyev, and Wei Song. The results are quite encouraging. Area under the curve is high. It's 90-97%. But what's more encouraging, especially given our history, we have been struggling to model enhancers for over a decade using SVMs, random forests, you name it. We barely pushed above 70-75%. And somehow this deep learning brings us to 95%, 90-95%. Okay, great, we'll take it. So what can we do with the method that's capable of predicting enhancers? Again, we're kind of interested in enhancers, but not that much. We are much more interested in mutations that disrupt enhancer activity. We can do enhancer mutagenesis. We can computationally make a mutation at every single nucleotide of a given enhancer and record whether a mutation is going to be damaging or not. And at the remaining slides, please keep in mind that whenever I'm going to be talking about a positive score, that's a score that has a negative effect on the enhancer activity. What was pleasantly surprising and quite unexpected when we looked at binding at transcription factors and we recorded these scores, mutational scores, what we saw is that they very nicely profile the binding specificity of transcription factors. And not only that, with the use of these methods, we can precisely establish boundaries of transcription factor binding sites, something that you can previously get with only ataxic footprinting or similar methods. And that was quite useful for us because we are going to use these binding sites to model the mutations. And how does it look when we apply these mutational profiles to enhancers? This is an enhancer. And what you see here, you see a few peaks, you see a couple of valleys. And when we look at what they correspond to known binding of H and F for alpha. Not only that, our method also predicts binding of other transcription factors, and we don't have to know what they are. We just know that they bind there, and we know that we can predict their location. Okay, that's fine, but what do we do with that? Again, we are interested in regulatory mutations. And there is a nice collection that has been published by Stencil's group 5 years ago of reporter assay QTLs. What they've done, they looked at the association of sequence variants with the level of reporter gene expression, and they found those that are in a strong correlation. When you look at the density of QTLs and enhancers, you see that it's high. Obviously, enhancers are functional. That's what you expect to see there. But what was encouraging is that the density at the predicted binding sites, according to our model, is much higher than the density of QTLs and enhancers. What's more encouraging, again, we are looking for these very short sequence variants, that the scores of our models at sequence variants, if they're high, they're much stronger associated with experimentally detected functional sequence variants. I told you about peaks and I told you about valleys. So there are some positions within the enhancers that somehow our model predicts to have strengthening effect on the enhancer activity. And that was kind of crazy to us. And it was quite not well received in the field until our collaborators from UC Irvine, Kwon's lab, and Len Pitakia's lab from Berkeley lab, what they've done, they took that ZRS enhancer that I mentioned previously, and they did the enhancer mutagenesis in vivo. And they looked at the change in expression pattern. And what was quite unexpected, I would say, is that 71% of those mutations that had a phenotypic effect, they were associated with enhancer strengthening, with gain of enhancer activity as opposed to deleterious effects. So we used that model in collaboration with Francis Collins' lab at the Genome Institute to apply to the study of type 2 diabetes and trying to identify causal mutations of this disease. So their lab had a collection of over 7,000 of islet enhancers. We applied our method, and we built a model. The first question that we asked whether this model works or not, and we used data from Kelly's lab that was published about 10 years ago, in which they recorded effects of sequence variants on the activity of the reporter gene, and they had positive and negative effects. And our model, as you can see there, in the vast majority of cases, correlates well what has been recorded experimentally. That's great, but how does it look in real life when you look at a single enhancer? And this is a correlation of what we predict with experimental data that was published by Akituv's lab about 5 years ago on a particular enhancer. What you see on top, these are the results of MPRA experiments. And as you see there are two tall spikes indicating two very particular regions within that enhancer that have very damaging effect on the enhancer activity if mutated. And underneath it, you see our computational predictions. And what you might notice is that exactly underneath these two spikes, there are two spikes of our model correlating really well with what experiments have been showing us. As a computational group, we have to correlate with other methods. On the right, you see the correlation of what we do with other deep learning methods, ensembles of deep learning methods, and our model performs quite well. And if you take SVMs into consideration, it performs really well. Okay, going back into type 2 diabetes. So how does GWAS look in this case? GWAS of type 2 diabetes brings you about 400 associations, 400 and some. If you extend those using LD blocks, you get to 12,000. Experimentally managing and sifting through this is quite challenging. That's why we still don't know everything about type 2 diabetes. But if you apply this method that I have been talking about, what you see is that we can narrow down the search space to just 52 SNPs. How does it look like when you look at a particular gene associated with type 2 diabetes, RNF6, for example? In the locus of this gene, you have an LD block of 30 SNPs, and somehow you need to resolve it. Our method says that one of them, the one that you see over there, the red one, is the causal SNP. And in this case, we were lucky because that SNP has been experimentally profiled and it has been shown to result, the variant at that position has been shown to be associated with a 6-fold decrease in the level of gene expression. So that was great. We looked at some other cases, and Stitzel's lab from Jackson's, what they've done, they said, okay, why don't we try to experimentally validate a few of the predictions that you are making? And this is the locus of PSMA1, another gene associated with type 2 diabetes. When you look on top at the GWAS associations, it's a mess. Glucose associations, next row, can help you tailor it down to a few SNPs, in this case, 3 SNPs. And out of them, they think that the one on the right is the causal. Our data says that the one in the middle is. So how do you resolve it? Again, we're not experimentalists. We are afraid of pipettes. So Michael's lab, what they've done this electromobility shift assays, and they did luciferase reporter assays, and they have been able to indeed demonstrate experimentally that the SNP in the middle is indeed the causal SNP, and 2 other ones are just neighboring SNPs. So do I have a minute? Am I out of time? Okay. So let me briefly tell you maybe a couple more minutes about another study that I think quite exciting. So with that method, we can apply it to studies of brain enhancers. We are looking at Alzheimer's, Parkinson's. We can look at developmental enhancers, et cetera, et cetera, et cetera. What is interesting is that we can model human enhancers and enhancers from other species. And in this case, we looked at human and chimp neocortex enhancers. And we looked at the evolution of neocortex. And we asked a very, very simple question. Have enhancers changing from one species to another, and how do you gain enhancers? What is associated with enhancer gains? How many mutations do you need to gain a new enhancer? In this case, in neocortex. And quite puzzling, interesting observation was 1. In 40% of enhancer gains, one mutation is sufficient to change neutral template into neocortex enhancers in humans. It might not be surprising to know that those mutations are associated with mathematical abilities in humans, with autism spectrum disorder, et cetera, et cetera, et cetera, et cetera. But when we looked at the strengths of the association, we have been quite puzzled and surprised to observe that the strengths of these associations at the mutations in these enhancer gains has been much, much, much stronger than at the evolutionarily conserved enhancers. And with that, I'm going to stop and tell you that this has been the work of the members of my lab, Dihuan Vaison, Sanjar Khodayberdiyev, Shan Li, as well as it relied heavily on our experimental collaborators, Francis Collins' group from NHGRI, and Pinakia's group from Berkeley, as well as several other labs. And thank you so much for your attention. And if there are any questions, I'd be happy to answer.
Do you answer questions? Yes.
Thank you. I wonder, have you tried changing the distance between the transfer factor and the keys, or switch all of them to see if that will improve the answer capability, as you just showed in silicon and Genesis?
So there has been a lot of discussion, right, in this gene regulatory fields about the encryption of gene regulatory elements, and the billboard model seems to be the prevailing mechanism of the structure of gene regulatory elements. We have a paper with Nada Vahitov that was published 8 years ago, in which we tried to create liver enhancers from scratch. We took a neutral template and started dropping in transcription factor binding sites and looking whether it's the position is important, the distance is important, the orientation is important. Nothing is important except the number of binding sites that you put in. It's kind of a soup model. If you get enough ingredients in, doesn't matter what. If it's a liver transcription factor binding site, you get an active enhancer. And that makes this modeling of enhancers extremely challenging because there are no strict rules in the non-coding parts of our genomes that would be easy to model computationally. I think that's the reason why all the SVMs and prior models have been failing so miserably in the past, and why this deep learning that we have no idea what it does so well. So I think the use of explainable AI is going to help us a lot in trying to understand this grammar of gene regulatory elements and try to understand this a little bit more if there are any kind of convincing rules depicting where this binding site should be, what is the distance between them, orientation, is to learn a little bit more about their structure.